Good morning to you all. Myself, I am Dr. M. Indira, Associate Professor in Department of Biotechnology, Vignan's Foundation for Science, Technology and Research, Andhra Pradesh, India. Along with my colleague Dr. K. Prakash Narayan Reddy, we both are giving a series of lectures related to the course Microbiology. Now we are in second unit, that is major groups of microorganisms. In the previous lecture, we covered the topic 1, that is Introduction to Diversity and Classification. Now we will see the second topic, that is characteristics used in taxonomy. So here in this unit, we will learn about how we can arrange the organism into a particular class, that means taxa, and what are the characteristic features used for identification and classification of the microorganisms. So in the previous class, we have seen what is taxonomy. It consists of identification, classification and nomenclature. So these three aspects are very much essential when we are identifying any organism, when we are placing any organism into a particular group. Okay, so for this, what are the characteristic features used for identifying any microorganism? This is a practical approach where we have to study about the external features as well as the internal features. Internal features means nothing but the genetic material, the proteins, all the genome, everything comes under the internal features. External features, morphological features. Okay, so all these practical approaches are required for classifying the organism into a particular group. Now we will see in this topic. The objective of this topic is to overview the concept of characteristic features used for identification of microorganisms. So that is the objective of this topic, and the expected outcome from this topic is the practical approaches for identification of microorganisms and classification of microorganisms will be acquainted. What are the practical tests used for identification as well as classification of microorganisms, we will learn from this topic. Now, the taxonomic characteristics. So here this taxonomic characteristic is nothing but expressed attribute of an organism that we have to evaluate. What is the characteristic feature? It is an expressed attribute. The taxonomic characteristic is nothing but it is an expressed attribute of an organism. It is going to be evaluated. Okay, so here what are those characteristic features? If you see here, there are classical characteristic features and molecular characteristic features. Classical characteristic features, if you see here, morphological features, biochemical features, physiological, metabolic, ecological and genetic. Morphological features, nothing but external features like cell shape, cell size, cell color, the microorganism color, how they are arranged. Okay, so all these comes under the morphological features. We will learn in detail in the next slides. Biochemical features. Biochemical features, nothing but the biochemical reactions taken place inside the living organism. So the substrate is converted into product by the action of enzymes. So this is a chemical reaction, biochemical reaction taking place inside the microorganism. So what are the biochemical tests used for identifying a particular microorganism that we will see. Next, physiological. Physiological is nothing but the environment, the mode of nutrition, the requirements of carbon, nitrogen, all these patterns we will learn under the physiological features. Metabolic features. Metabolic features are nothing but the enzymes involved in the biochemical reactions and the metabolites produced from the microorganisms. Primary metabolites are there, secondary metabolites are there. What are the metabolic reactions and what are the metabolites produced from an organism? That we will evaluate under metabolic. Ecological features. Ecological features nothing but the microorganism and its surrounding environment. Its surrounding environment means neighboring microorganisms. It may be a plant, animals or human beings. So that is the ecological features. And genetic. What is the genetic material? What is the genome? Genome size of the organism that we will use for identifying the organism. And next, if you see the molecular characteristic features, it's related to nucleic acid base composition. So we all know that nucleic acids, genetic material, DNA, RNA. So what is the nucleic acid base composition? And next, nucleic acid hybridization, nucleic acid sequencing, genome fingerprinting and amino acid sequencing. So these are the molecular characteristic features and classical characteristic features we will learn for identification as well as classification of microorganisms. See here classical characteristic features, morphological, physiological, biochemical, ecological and genetic. We will see one by one. Now see the morphological features. If you see the morphological features, for example here I have shown some of the morphological features in the picture. See here Vibrio cholera shape, bacillus structure, streptococcal, diplococci and then staphylococcus, bunch type of the organisms. Here you can observe comma shape, then bacillus type, a chain, streptococcus a chain, bunch type of, as well as here spirochetes, spirillum type, like this. So these are the external morphological features. They are used for identifying the organism. So not only the shapes, next cell size. What is the size of the organism? Whether it is in micron size or meter size. Generally microorganisms are in micrometers. Okay. So next cilia and flagella. These are the external features located on the surface of a bacterial cell. For example, this is a bacterial cell. So flagella structures, cilia structures like this. So those are the features used for identification of the microorganism. Cellular inclusions. Cellular inclusions are nothing but insoluble protein molecules, or we can say the storage reserves present inside the bacterial cell, like this. So those are called cellular inclusions. And another morphological feature, color. If you see the microorganism, what is the color of the organism? Whether it is pink color, yellow color, white in color or red color. So based on the color, we can identify the organisms. And next, another feature is the mechanism for motility. Some of the microorganisms, they are motile in nature, and some of the microorganisms, they are non-motile in nature. So based on the movement, the microorganisms, we can identify whether it is motile or non-motile. And And next, another feature is the endospore. Endospore and location. If you see the endospore, endospores are nothing but dormant structures whenever any conditions are unfavorable. So, the bacterial cell forms the endospore structures. Those are the dormant structures, and whenever the conditions are favorable, then this endospore bacterial cell turns into a vegetative cell and starts its progeny. That means replication and reproduction. Okay. So the location also, whether it is located central or peripheral. Next, colonial morphology. So whether the microorganism is transparent in color or it is opaque. So those features used for identifying the external features of microorganism. And ultrastructural features as well as staining patterns. So for example, staining. Why we will use staining? For identifying the organism. Generally the microorganism is, if it is not stained, we cannot see it under the microscope. So we will use particular dye. Color compound is given to the microbial cell, then it appears in color so that we can observe under the microscope. So again in this gram positive, gram negative. Like this, we can observe the staining patterns. Using those staining patterns, we can identify the microorganisms. So all these are the external morphological features. The external morphological features used for various groups of microorganisms. Cell shape, size, colonial morphology is used for all major groups of microorganisms. And staining behavior used for bacteria and some fungi only. Next, cilia and flagella, so all major groups of microorganisms. And then motility, endospore. Here endospore forming bacteria. Some of the bacteria, for example, if you take bacillus, this bacillus produces the endospores. Another genus, that is Clostridium, it produces endospores. So we can differentiate. For example, if we isolate any organism, we can identify based on the staining pattern whether it is producing any endospore or not. Dyes are used for identifying the endospore. If it is producing, we can categorize this particular organism into a endospore form. If it is not producing, we can arrange that organism into a non-endospore form. Like this, we can identify the organisms. Next, physiological metabolic characteristics. Here, if you can observe the physiological patterns, the general nutritional type. So what is the food habit of a particular organism? What nutrition it is consuming? For example, if you take ourselves, some people may like chapati, some people like biryani. That means the food habits differ from one another. Like this, the microorganisms, the nutritional pattern also varies based on the nutritional type, whether it is autotrophic or heterotrophic, whether it is chemotrophic or organochemotrophic. That means what is the energy source and what is the light source and what is the carbon source for a particular organism to synthesize a food. So based on this nutrition, we can classify the organisms into different groups. Okay. And next, carbon and nitrogen source. If you see here, carbon source nothing but carbohydrates, lipids, hydrocarbons, all these contain carbon. And inorganic carbon, nothing but carbon dioxide. If you see here, the microorganism, whether it is utilizing inorganic carbon or organic carbon. If it is utilizing organic carbon, again in that which organic compound it is using. It is a carbohydrate or it is a lipid or it is a hydrocarbon. And again if you take carbohydrates category, which carbohydrate it is utilizing, whether it is glucose, lactose or sucrose. Like this, the microorganisms, their requirement, carbon requirement varies from one organism to another organism. Similarly, the nitrogen also, whether it is inorganic nitrogen or organic nitrogen. And next, cell wall constituents. Here we can differentiate microorganisms into two types, gram positive and gram negative. Gram positive cell walls consists of peptidoglycan layer and teichoic acid, whereas gram negative cell walls consists of lipid layer, more amount of lipid layer and very less amount of peptidoglycan. So based on the cell wall composition, we can differentiate one organism from another organism. Next, energy sources. Already mentioned, whether it is using light energy or chemical energy. And conversions. Next, fermentation products. Any microorganism utilizes the food material and starts undergoing metabolic reactions. So in this metabolic process, it converts one form of the substrate into another product. Those products are nothing but metabolites. So microorganism produces primary metabolites, secondary metabolites. So what are those metabolic compounds produced by an organism? For example, say microorganism utilizing glucose. So after fermentation, fermentation is nothing but metabolic reactions, metabolic pathway. So after utilizing this glucose, the glucose is converted into alcohol or pyruvic acid or acetic acid. You will learn in the biochemistry also. That means the microorganisms convert one form of the substrate into another product. Luminescence, it's related to emission or absorption of light. Motility, whether it is motile or non-motile. Osmotic tolerance. Some of the organisms, they can tolerate the high salt concentration. So some organisms, they cannot tolerate the salt concentration. So based on the osmotic tolerance, we can differentiate the organisms. And storage inclusions already mentioned. So nothing but insoluble protein molecules, these are the energy reserves present inside the cell. Some bacteria or some microorganisms, they produce insoluble molecules. And next see is another feature used, that is the temperature as well as pH. If you see here the temperature, the microorganisms requires different temperatures. For example, say microorganisms located or present in the hot environments, they are able to grow at high temperature. Some of the microorganisms, for example, if we take a refrigerator, so refrigerator inside the microbes will grow. That means the temperature inside the refrigerator is at very low temperature. For example, say 10 degrees. There also the microorganisms are growing. If you see another example, if you take our body also consists of microorganisms. Our body temperature is normal room temperature 37 degrees centigrade. So where the microorganisms live in our intestine and the gut, that means they are tolerating the normal room temperature 37 degrees centigrade. That means the microorganisms, their growing under various temperatures differs from one organism to another organism. So based on the temperature requirement, we can differentiate the organisms and we can identify the organisms. Similarly, the pH also. Some microorganisms, they grow at very low pH, acidic pH. For example, say if you take stomach. Stomach is acidic in nature. There the microorganisms present inside our body in the stomach, they grow at acidic rate. If you take intestine, where the pH conditions are alkaline in nature, those microorganisms present in the intestine, they are growing at alkaline conditions. So the microorganisms, based on the pH, we can differentiate from one another. Next, photosynthetic pigments. Some of the bacteria, they are having photosynthetic pigments. That means they are having the ability to synthesize the food material. Next, salt requirement. For example, if you take the fresh water or drinking water consists of microorganisms. Those microorganisms, their salt requirement is very less. If you see the marine environment, oceans, seas, where the salt concentration is high, there also the microorganisms are present. If you compare this fresh water and marine water, the freshwater microorganisms, their requirement is very low salt concentration, and marine organisms, their salt concentration is higher. So like this, we can differentiate the organisms. Secondary metabolites. So I mentioned under fermentation products, some of the microorganisms, they are having the ability to produce secondary metabolites. For example, if you take antibiotic. Antibiotic is produced by the microorganism, you all know about it, that is Penicillium notatum. The organism having the ability to produce the metabolite, that is secondary metabolite, antibiotic. Some organisms, they may not produce metabolites, secondary metabolites. So like this, we can identify whether the organism is producing any secondary metabolite or not. Sensitive to metabolic inhibitors and antibiotics. Some microorganisms, they are sensitive to inhibitors. For example, if you take antibiotics, some organisms, they are sensitive to antibiotic, and some organisms, they are resistant to antibiotic. So, like this, the physiological and metabolic features are used for identification and differentiating the organism into a particular group. Next one is the ecological and biochemical characteristics. Ecology is nothing but the organism and its surrounding environment. So the interaction between the organism and its surrounding environment is called ecological interactions, where we can observe the interaction of microorganisms with other organisms or with other animals, or we can study the life cycle pattern of a particular organism. How it is multiplying, what are the conditions, what is the habitat, everything comes under the life cycle pattern. And another one, symbiotic relationship. The microorganisms, they are symbiotic with the other organisms. For example, say plants, animals and humans. Even in our body also, the microorganisms present inside our gut, they are symbiotic. Okay. And some organisms, they are symbiotic with the host also. That means they produce some compounds. Those compounds are utilized by the host cells. And some host cells, they produce some proteins or compounds. Those are taken up by the microorganism. Like this, the symbiotic relationship will be maintained for an organism in their surrounding environment. And ability to cause the disease. Some of the organisms, they are harmful, pathogenic in nature. They cause the diseases. Example, if you take E. coli. E. coli is present in the water bodies. When we consume any normal water or polluted water, the microbes entered into our body and causes the disease. Next, habitat preferences, temperature, pH, oxygen and osmotic concentration. So the microorganism, for growth, it requires certain temperature, certain pH, salt concentration and osmotic tolerance. So these habitat environmental conditions are very essential for growth of an organism. So these patterns are used for identifying the ecological behavior of a particular organism. And next we will see the biochemical characteristics. Here it's a totally practical approach. In our microbiology lab also we will perform these experiments for identifying the organisms. If we see here, fermentation test, gelatin liquefaction, hydrolysis, litmus test, catalase, enzymes, coagulase, oxidase. So these are the enzymatic tests used for identifying the metabolic pattern of the organism. And this is the another IMPIC test. Indole, methyl red, Voges-Proskar and citrate utilization test. So these tests are used for differentiating the enterobacteria. What are these enterobacteria? Enterobacteria are nothing but those organisms present inside our intestine. So in the intestine, if any bacterial infections are identified, then which organism is causing a particular infection? We can identify, we can differentiate based on this IMPIC test. So in clinical labs, we can use this test for differentiating the disease causing organism. Another hydrogen sulfide test, phenylalanine and urease test. So here in the biochemical characteristics, we can study the biochemical reactions. Biochemical reactions. Those biochemical reactions are taken place by the enzyme. So the organism is able to produce a particular enzyme or not. For example, take catalase. If the organism is producing catalase enzyme, we can say the organism is catalase positive. If it is not producing, we can say that is catalase negative organism. Similarly, oxidase, coagulase, like this, if they are producing any enzyme, we can say enzyme producers. If they are not producing enzymes, non-producers of particular enzyme. So like this, the ecological and biochemical characteristic features are used for identification and classification of the organism. Next, another characteristic feature and classical characteristics is the genetic analysis. This is the old approach where the scientists used for identification and classifying the organisms. They used the transformation pattern of the organism as well as the conjugation pattern of the organism for taxonomic identification. For example, if we take E. coli. So this E. coli, it undergoes conjugation with the another species that is Salmonella and Shigella. But it not with the organism Proteus and Enterobacter. So these organisms, they belongs to the category that is Enterobacteriaceae, family as well as order Enterobacterians. Here you can observe the E. coli undergoes conjugation with these species Salmonella and Shigella only, but not with the Proteus and Enterobacter. So for this conjugation and transformation, the plasmids are used for analyzing the phenotypic traits. So this is the old approach used for identification and classification of the microorganisms. But now, with the advancement of technology as well as the tools, instruments and techniques, now we are in a position to identify the genetic material, that is the DNA, RNA. The genetic material, what is the sequence of the genetic material, what are the proteins present in the cell, we can able to identify. Okay. Now, we have discussed classical characteristic features: morphological, physiological, metabolic, ecological, biochemical and genetic analysis. Now we will see the molecular characteristics of an organism. There we can study the nucleic acid base composition, nucleic acid hybridization, sequencing, genomic fingerprinting and amino acid sequencing. We will see one by one. Now see the nucleic acid base composition. What is nucleic acid? Nucleic acid is nothing but the genetic material, either DNA or RNA. Okay. So if you see the genetic material DNA, it consists of 4 nucleotides. What are those nucleotides? Guanine, cytosine, adenine and thymine. You all know about these 4 nucleotides: guanine, cytosine, adenine and thymine. These are the 4 nucleotides present in the genetic material DNA. Whereas in RNA, uracil, thymidine is replaced by uracil. Okay, see here GCAT. So these nucleotides are used for identifying the base composition. How much amount of guanine and cytosine content present inside the genetic material. This is the formula used for calculating the nucleic acid base composition, where guanine and cytosine content by total nucleotides, guanine, cytosine, adenine and thymine. So total content into 100. This gives the nucleic acid base composition percentage of the guanine and cytosine. Okay. So higher the G plus C gives the higher melting temperature. Here if you observe, G always binds with C. A always binds with T. If you see the bond between these nucleotides, here triple bond. For breaking this triple bond we require higher temperature. Whereas in this case only double bond. Breaking this double bond requires very low temperature. So higher the G plus C content gives the higher melting temperature. Next, molecular analysis. This G plus C is also an historical old approach. Now that is the hybridization, sequencing and amino acid sequencing methods. Now we will see the nucleic acid hybridization. So nucleic acid hybridization, based on this how we can identify an organism and how we can arrange this organism into a particular group. That means similarity nature. Here you can observe organism A, genetic material, and organism B, genetic material. So the genetic material, nothing but DNA we have to take, and separately we have to melt the genetic material. Heat to separate is nothing but melting the genetic material. So whenever we melt the genetic material double strand broken down into single strand. Next, mix these strands in a test tube. So if these two organisms they are similar, then after cooling the temperature they will hybridize. Here you can observe the result like this. If you see the first case where the degree of hybridization is 100%, that means these two organisms are closely related. That means the genetic material is similar. So the hybridization is 100% and we can say that these two organisms are in one particular group. If we see second case, partial hybridization, that means some amount of the nucleotides, they are not complementarity with the another strand. So we can observe the partial hybridization. See here the nucleotides are not complementarity with each other. So that means these two organisms, they are differing a particular portion. And the third case, no hybridization. That means these nucleotides from one strand to another strand, they are differ from one another. They are not complementarity to each other. So we can observe the no hybridization. That means these two organisms A and B, they are not similar. So like this, the DNA hybridization technique is used for arranging the organism into a particular group based on the similarity, complementarity of the gene. These are the steps. First step, heat the mixture of the double strand, then separate the single strands. Next, third one, mix those two organisms single strand, and later cool the test tube. That means reduce the temperature, and then we can observe the annealing, that is the hybridization. Based on the hybridization, we can represent like this. So here, if same species, their hybridization is 75 to 100%. If the species are similar in nature, their hybridization 75 to 100%. And if two organisms are taken, if the hybridization is under 25 to here 75, then we can say those two organisms are similar but comes under one particular genera. And next, 25% of hybridization, that means those two organisms are differ and they are from one genera to another genera. That means the genus is differ for each two organisms. If the two organisms are similar, they come under same species. For example, say Bacillus cereus, Bacillus subtilis. So here if you take Bacillus cereus, again in the two species, that means they are called as strains, type strains. They are similar, comes under the same species. And next, same genus but different species. Same genus means example Bacillus cereus, Bacillus subtilis. Cereus is another species, subtilis is another species. But these two species comes under the genus Bacillus. Different genera. One is the Bacillus genera and another one is the Clostridium. So like this, the organisms are arranged into a particular group. Next, another one is the nucleic acid sequencing. Here, the genetic material, the DNA, what is the genome size, what is the nucleotides present in the genome, we can sequence with the help of nucleic acid sequencer, nothing but DNA sequencer. For example, if you take here microorganisms, in this case, we will use this 16S. We will use 16S rRNA sequencing in case of prokaryotic organisms. In case of eukaryotic organisms, 18S rRNA is used. For microorganisms, here 16S rRNA. If you see here, microbes consist of 70S ribosome. Eukaryotes consist of 80S ribosome. In the 70S ribosome, this consists of two subunits. One is the smaller subunit and another one is the larger subunit. The smaller subunit is encodes 21 proteins and it is nothing but 16S rRNA. This is used for identification identification and classifying the microorganisms based on this 16S rRNA sequencing. And if you see the larger subunit, that is 50S, and it is made up of two subunits. One is the 23S rRNA and another one is the 5S rRNA. If you see these three units, this 16S rRNA is used. Why? Because this 16S rRNA genetic material consists of conserved regions. Throughout the generations, the genetic material is stable. So, those conserved regions are used for identification and classification of the organism. So, this 16S rRNA sequencing is used for identifying the organism. After identification, using bioinformatic tools, we can place this organism under a particular group, which genus it belongs. We can perform the bioinformatic tools nothing but Clustal W, phylogenetic relationship. That means we can arrange this organism into a particular group. So this is the 16S rRNA is used for identifying the organisms. This is the pictorial representation of sequencing. So you see here you can observe GT like this. That means the first nucleotide to last nucleotide, the nucleic acid sequencer gives the entire genetic information, nucleotides information. So this sequence data like this in the form of FASTA file. So this FASTA file data, using bioinformatic tools, we can identify the phylogenetic relationship of this particular organism with the neighboring organisms or similar group of organisms. Next, genomic fingerprinting. So this is the another technique used for microbial classification and we can determine the phylogenetic relationships. Here in this genomic fingerprinting, it does not involve the nucleotide sequencing. Here we will do the polymerase chain reaction, that is the amplification of the genetic material. Okay, I will show you the diagrammatic representation. Here you can see, for example, if you want to identify an organism present in our mouth, saliva, okay, that we have to take and grow in the laboratory. So we can observe the microorganisms that we have to take. Next step is the isolation of the genetic material, that is DNA, and this DNA is amplified using PCR, polymerase chain reaction. After amplification, then the sample is run under electrophoresis. So this electrophoresis gives the different band patterns, image like this. Each and every band represents one particular organism genetic material. So this pattern is analyzed like this. Using software tools, we can arrange a particular organism, whatever we isolated. We can arrange that organism into a particular group. This is the phylogenetic relationship. When we perform phylogenetic analysis, that is Clustal W, we can get the phylogenetic tree like this. So, the genomic fingerprinting, where the polymerase chain reaction is used. After amplification, the sample is analyzed using electrophoresis techniques and the band pattern is analyzed by bioinformatic tools. Next, amino acid sequencing. So far we discussed genetic material DNA. Now the proteins. The DNA encodes the proteins. Proteins are made up of amino acids. Okay. See here, the amino acid sequencing. What is the first amino acid, second, third, like this. The entire protein sequencing data will be obtained by amino acid sequencer. So these are the advanced technologies. Here, so the amino acid sequencing of some proteins, they are used for identification and phylogenetic relationship of the organisms. Cytochromes, histones and heat shock proteins. So, based on the presence and absence of these proteins, we can identify the organism and we can see the phylogenetic relationship with the other organisms. Now the summary of this topic. Here the characteristic features used for identifying the organism. So classical characteristics and molecular characteristics. So classical characteristics based on the phenotype. Here you can observe the phenotype, external features. Molecular characteristics based on the genetic material, where the DNA hybridization we can use, nucleotide sequencing we can use, amino acid sequencing is used for identification and classification of the organisms. So after going through this topic, so you all have to read these questions. So refer the textbooks and study these questions. First one is the what are the classical characteristic features, describe briefly. Enlist the biochemical and ecological characteristic features. Enlist the molecular characteristic features used for taxonomy and describe the 16S rRNA sequencing. This is very important for identification and classification of microorganisms. So in the previous class also we discussed 3-domain classification where the microbes classified into bacteria, archaea and eukarya. So after the advancement of the technology, the scientists use 16S rRNA sequencing. So this is the phylogenetic relationship where we can identify the microorganisms. Next, write the steps involved in DNA hybridization with neat sketch and draw the genomic fingerprinting flowchart for identification of the microorganisms. Study all these questions and prepare for exam. Thank you all.